Alrighty. Good afternoon, everyone. This is likely probably your last breakout session for the week, so thank you guys for joining us. Today we have three individuals that came all the way from Japan to be here today. We have Yoshitaka Yuki and Tajuki from Shionogi, and they're here today to tell us about overcoming challenges in real-world evidence generation, the AI SaaS for RWE approach. And we'll save time at the end for any questions.
Yeah. Thank you for my introduction. So, hello everyone. So, thank you for giving us such an opportunity to speak about AI SaaS for real-world evidence and our data science activities today. I am Yoshitake Kitanishi, the head of data science department from Shionogi. Today we will be speaking on overcoming challenges in real-world evidence generation, the AI SaaS for real-world evidence approach. Firstly, let me briefly introduce our company Shionogi. Shionogi is a Japanese pharmaceutical company founded in 1878. Of course, we also have a group company, Shionogi Inc., in the US. Although this is a drug discovery-oriented pharmaceutical company, its vision for 2030, building innovation platforms to shape the future of healthcare. And it is also focusing on creating a variety of services, including AI SaaS for real-world evidence, which we introduced today. Let me start by introducing our data science department. As you can see, it is positioned within the DX promotion division, along with the IT department. The data science department and IT department work very closely together to utilize internal and external data to achieve various digital transformations. The close cooperation between these two departments is key factor for the success of DX. Our data science activities are closely linked to value chain. In fact, value chain in the company generates a large amount of data every day. For example, there is self-imaging data in research, clinical trial data in development, sales activity data in sales and marketing, HR skills and employee engagement data in human resources, accounting data in accounting and finance, and so on. We are covering the entire value chain and cross-functional activities. Now, I would like to share with you over 20 years of history with SaaS products. Shionogi's data science activities have always been with SaaS. This is because SaaS is a golden standard system that should be placed at the center of the analysis infrastructure. Having cultivated a solid reputation for the comprehensiveness of its analysis methods and the accuracy of its analysis results, we started with PC SaaS, SaaS 6, 8, and then moved on to PH Clinical and SaaS drug development and SaaS clinical data integration, Global SaaS Server, SaaS 9, SaaS IMStat, and the SaaS Data Loader, and the SaaS Visual Analytics. Recently, we built the SaaS barrier on AWS with Snowflake architecture and further updated the environment. As you can see, SaaS barrier is positioned as the score system. SaaS barrier and Snowflake work together to dramatically accelerate the speed of analysis. We would love to share this story with you sometime. So my part is finished, and the next presenter, Yuki Yoshida.
Hi, hello. My name is Yuki Yoshida. Before introduce our AI SaaS for real-world evidence, I want to share AI SaaS programmer system with you. That is the foundation for the development of AI SaaS for real-world evidence that we are today presenting. This is our history. In 2018, it was a significant turning point for our transformation. We released the AI SaaS programmer system based on SaaS Buyer in 2018. AI SaaS is the first system in the pharmaceutical industry to create SaaS programs semi-automatically using AI technology. With the release of the AI SaaS, Shionogi has achieved business efficiency. That experience changed our mind for working efficiently using AI. Let me explain the flow of the analysis in the clinical trials and describe what the AI SaaS generates. Today we introduce AI SaaS for real-world evidence, but we previously worked for analyzing the data from clinical trial. That is an important part of our work. So at that time, in the clinical trial, we usually plan hundreds of TLFs, as you may know, for demographic efficacy safety analysis. We need to make SaaS programs for each analysis, as you know, so we have to create hundreds of SaaS programs for only one clinical trial. Then we collect the data and make metadata according to CDISC standards as well. The AI SaaS create these SaaS programs semi-automatically. AI SaaS follows validation analysis of clinical trial, the flow, and output. That means the AI SaaS did not only achieve efficiency, but also controlling the quality of the output. Here, I show the concept of AI SaaS. AI SaaS has 3 steps: recognition, learning, action steps. First, in recognition step, collect necessary information from documents of target clinical trials. That is here. And the next, in learning step, predict and search appropriate information from the past clinical trial data or metadata. Finally, in action step, AI SaaS semi-automatically outputs SaaS programs. We use mainly CNN technology for the system. That is the overview of the AI SaaS. We started the project from 2016 using the SaaS AI version 2 here. We achieved 33% business improvement in 2018. We could reduce the standard programming time by 100 hours. the AI SaaS was evaluated by outside of my company, and we got a first place winner of the Innovate Problem Solver at the last year of the SaaS Innovate. So that was our history, and we are leveraging the SaaS AI as a starting point to further streamline our business processes. As part of this effort, we will introduce further streaming of the broader clinical trial process and today's main topic, expanding AI SaaS to real-world evidence. As you know, there are a lot of steps in the clinical trial to complete CTD. AI SaaS is a system designed to streamline the analysis process here. However, as I said, when we look at the entire clinical trial process, we see that before the analysis stage, where the SAP is created, there is a protocol development stage as well. Similarly, after the TLF generated, there is a task of writing CSR. Now, we are trying to leverage generative AI technologies to streamline the entire whole workflow from upstream processes like protocol generation to downstream processes like CSR generation in end-to-end manner. At Shionogi's data science department, as Yoshitake showed, we cover all processes, so we have already started working together with medical writing department. When we break down medical writing activities into documentation, review, and finalization, we are currently focusing on documentation stage. We are confident that that initiative will lead greater efficiency. We have successfully developed an application using Streamlit that can generate CSRs. My colleague Tadashi Matsuno will be presenting the whole detail at PharmaSag in June. So if you go to visit the PharmaSag, please take a look at his presentation. I encourage you to attend the presentation. So that is our basic consideration, and now I want to expand this concept to the real-world evidence. AI SaaS for real-world evidence is an innovation that expands the successful process improvement we achieved in clinical trials using AI SaaS and generative AI to the analysis of real-world data. My colleague Takuji will introduce you to the whole story. So I want to ask Takuji to present the stories. That's all from me.
Okay, now Takuji Komeda will take over the presentation. I usually work as a data scientist, engaging in tasks related to the creation of real-world evidence using real-world data. I would like to introduce expanding AI SaaS to support real-world data analysis programming. Sorry. As previously explained, AI SAS has streamlined analysis-related tasks in clinical trials. On the other hand, there has also been an increase in the generation of real-world evidence from analysis of real-world data outside of clinical trials. Here, we present the result of a survey that examined a number of Japanese RWD articles in PubMed. Considering that 2021 data only covers up to June, it shows that RWD analysis has been becoming more active in recent years. Therefore, we believe that in addition to clinical trial analysis, improving the efficiency of RWD analysis is also an issue that needs to be addressed. On the other hand, the transparency of RWD research is sometimes questioned. Here, we present a schematic flow from obtaining RWD to publication of papers. Research institute, including pharmaceutical companies, commercially obtain RWD from RWD vendors. Subsequently, researchers within these institutes are expected to create protocols and analysis materials and conduct analysis based on them. The analysis results are then submitted and published as papers on journals. Unlike clinical trials, the processes of obtaining data is not subject to review by ethics committees. So large amount of RWD can be obtained and analyzed through commercial license agreements without going through an ethical review committee. Additionally, unlike clinical trials, there is no requirement to submit a protocol or analysis documents to ethics committee or regulatory authorities. In addition, most journals do not require submission of the actual data or code used for analysis. Furthermore, within research institutions, as long as one has access to the data, the one researcher can handle all processes from analysis to publication independently. Therefore, there is little third-party oversight, and it cannot be denied that the research method may have been adjusted to obtain results that were favorable to the researchers. Given this background, we believe that making RWD analysis more efficient through a transparent flow is necessary for accelerating RWD generation and improving its quality. Therefore, we began work on developing AI SAS for RWE, which applies and expands AI SAS for clinical trials to RWD. However, due to differences in data format, analysis tasks, and scope of issues to be addressed, simply applying the conventional AI SAS to RWD will not realize AI SAS for RWE. Here, some examples. In clinical trials, raw data formats are standardized by CDISC, but in RWD, the formats depend on the vendor. Therefore, due to these different format differences, the same processes cannot be applied to the entire RWD. Additionally, while clinical trials typically involve at most several thousand cases, RWD database can exceed 10 million cases. Consequently, since analysis variables are created from a huge number of records, a large amount of resources are also allocated to creating the analysis dataset, ADS. Regarding analysis documents, clinical trials are regulated by GCP SOPs, but in RWD, the extent and format of document creation depend on the project. This leads to challenges in organizing training data. In terms of analysis methods, RCTs do not have confounding, but RWD, being non-interventional observational data, does. Therefore, analysis method and forms that consider confounding factors are necessary. To address these differences in the data format, analysis tasks, and scope of issues, we believe it is necessary to advance the approach of RWD standardization common data model, which organizes the format of raw data from each vendor, and semi-automation of RWD analysis, which makes the entire analysis process, including ADS creation, more efficient and transparent. This is the concept of AI SAS for RWE. In the approach of RWD standardization, we standardize the data structure of RWD from various vendors stored on Snowflake and convert it into a common data model. In approach of semi-automation of RWD analysis, we plan to extend the function of AI SAS to semi-automate the processes from a research question to a protocol, from the protocol to analysis documents, and from these analysis documents to ADS and TFL creation programs, along with automatic recording of this creation history. Standardizing RWD and using an extended AI SAS for semi-automated analysis and recording will streamline RWD analysis, accelerating RWE creation and improving its quality. From this slide, I will explain the detail of each approach. First, let me explain the approach to RWD standardization. First, when considering the standardization of RWD, it was necessary to decide which format it based it on. And this slide shows several typical standardization formats. Each has its own characteristics, but since Shionogi uses RWD from all over the world, we decided to refer OMOP CDM with a view to the possibility of expanding code conversion in the future. Let me explain the concept of RWD standardization approach. As previously mentioned, the data structure provided by RWD vendors varies, so it is necessary to convert them into standardized structured data. For example, column names for patient ID or prescription date differ among vendors. Additionally, date formats can vary. Without standardization, it is impossible to perform analysis using common programs. Therefore, standardization involves unifying these column names and formats. This standardization is based on OMOP CDM, as previously mentioned. However, regarding codes, Japanese RWD are difficult to convert to OMOP CDM. So while standardizing column names, we did not do code conversion. It means that Shionogi defined original CDM for our internal use based on the OMOP CDM. Regarding the approach of semi-automation of RWD analysis, we are planning a step-by-step implementation. Ultimately, by leveraging AI, we aim to semi-automate the processes from research question to a protocol, from the protocol to the analysis documents, and from these documents to SAS programs. Additionally, since we believe that the efficiency and quality improvement of RWD analysis is a challenge for the entire pharmaceutical industry, we are considering developing an application to enhance usability for more users. However, as previously mentioned, the type and format of analysis documents created in RWD analysis vary depending on the studies. And we believe that there is currently insufficient training data to integrate AI SAS. On the other hand, the efficiency of RWD analysis is urgently needed. Therefore, as a step one, we decided to take one approach to semi-automate the analysis process using rule-based, macro-based methods. Although detail will be explained later, in the step one, we aim to implement a rule-based semi-automated flow where analysis documents are created from a concept sheet with simple analysis conditions, and the programs are generated based on these analysis documents. This is a step one implementation image. First, researchers create a concept sheet that lists simple analysis conditions such as target population, exposure group, comparison group, outcomes, background factors, and analysis methods. For the variables listed in the concept sheet, the variable definition knowledge database is referenced. And if the corresponding variables exist, an ADS spec reflecting them will be created. Researchers input any additional variables or flags that need to be defined on the ADS spec. If new variable definitions are made, they will be accumulated in the knowledge database. Based on the created ADS spec and the analysis method on the concept sheet, TFL spec will be created using rule-based methods. Using the contents of ADS spec and TFL spec, SAS programs are generated, and ADS and figures are output. Finally, each analysis condition and results combined and saved as a report. These processes are recorded in real time using Git. By implementing this flow, each spec, analysis, ADS, TFL output, and report are semi-automatically created from a concept sheet that describes simple analysis conditions. Each process is automatically reflected in Git. Currently, the development of these series of functions has been completed. I would like to demonstrate these functions. First, I will show the demonstration of creating ADS spec and TFL spec from the concept sheet. Okay. This is a recorded video. First, researchers create a concept sheet. This concept sheet includes basic information for analysis, such as the target population, exposure group, comparison group, evaluated events, background factors to be adjusted, and analysis methods. Based on the concept sheet, an ADS spec will be created. At this time, the knowledge database, which accumulated past variable definition, is referenced. This is the outputted ADS spec. Although some parts require editing by researchers, the base variables for the analysis are outputted based on the concept sheet. As explained earlier, by referencing the knowledge database, code for variables previously defined are automatically inputted. After creating the ADS spec, TFL spec is created from the concept sheet and ADS spec. Please wait. The TFL spec includes the necessary tables and figures derived from the concept sheet, along with variables required to create these tables and figures. In this case, since IPTW was selected as the analysis method in the concept sheet, tables evaluating the eligible exclusion number, the distribution of propensity score, demographics, and incidence of events before and after IPTW adjustment are selected. Next, I will demonstrate the part where programs are generated from these specs to create a dataset, tables and figures and reports. This program generates a program to create ADS from the ADS spec. Please wait. Okay. Yeah. It's outputted. For variables that are not automatically created, nodes are provided in the programs. Researchers can add codes to the program based on the nodes. By executing this, ADS will be created. As a side note, in the demonstrations, we have removed the execution time as needed. as you can see, ADS has been created based on the ADS spec. Next, this program generates programs to output figures and tables from the TFL spec. In this case, table 1, 2, 3, and figure 1 programs are outputted. By executing these, the respective figures and tables are created. In this case, by executing table 1 SAS programs, table of eligible exclusion numbers is created like this. Finally, we compile all documents and analysis results into one file and output it as a report. This file. As you can see, the created report file contains the concept sheet, ADS spec, TFL spec, and figures and tables. Each time a program is executed, the productions are reflected on Git, keeping a record of when and what results were output. This is the last slide. To reiterate at Shionogi, by standardizing RWD and using an extended AI SAS for semi-automated analysis and recording, we will aim to streamline RWD analysis, accelerating RWE creation, and improving quality. That concludes the presentation. Thank you for your attention.
And then at this time, if you want to take any questions.
So when you say AI SAS, what exactly does that mean? Is it one of the, I think I saw SAS Studio over there. I'm kind of a little lost there, sorry.
Sorry, one more.
So what SAS product is this when you say AI SAS?
Yeah, AI SAS is our original system, and it was created using SAS Buyer on SAS Buyer.
Analyzing the RWD. How are you doing that within? Is it something that you are doing it manually?
Not manually, but this is also created by ourselves.
Okay.
Is everything written in SAS, or do you write in a different language to do the AI section?
A different language?
Currently, in the Snowflake part, it's out of SAS, right?
Yeah.
But the right side, it's on SAS.
Impressive.
Thank you.
Okay, thank you guys very much. Wonderful.
Thank you.